Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Tentacruel and its relationship with all animal Pokemon as they are all part of the Animalia clade. Ready? Let's go! Animals are multicellular, eukaryotic organisms in the biological kingdom Animalia with few exceptions. Animals consume organic material, breathe oxygen, have myocytes and are able to move, can reproduce sexually, and grow from a hollow sphere of cells, the blastula, during embryonic development. Animals form a clade, meaning that they arose from a single common ancestor. Over 1.5 million living animal species have been described, of which around 1.05 million are insects, over 85,000 are mollusks, and around 65,000 are vertebrates. It has been estimated there are as many as 7.77 million animal species on Earth. Animal body lengths range from 8.5 micrometers, 0.00033 in, to 33.6 meters, 110 feet. They have complex ecologies and interactions with each other and their environments, forming intricate food webs. The scientific study of animals is known as zoology, and the study of animal behavior is known as ethology. The animal kingdom is divided into 5 infrakingdoms/superphyla, namely Porifera, Ctenophora, Placozoa, Cnidaria, and Bilateria. Most living animal species belong to the infrakingdom Bilateria, a highly proliferative clade whose members have a bilaterally symmetric and significantly cephalized body plan, and the vast majority of bilaterians belong to 2 large superphyla, the protostomes, which includes organisms such as arthropods, mollusks, flatworms, annelids, and nematodes, and the deuterostomes, which include echinoderms, hemichordates and chordates, the latter of which contains the vertebrates. The much smaller basal phylum Xenocoelomorpha have an uncertain position within Bilateria. Animals first appeared in the fossil record in the late Cryogenian period and diversified in the subsequent Ediacaran period in what is known as the Avalon explosion. Earlier evidence of animals is still controversial. The sponge-like organism Otovia has been dated back to the Tunian period at the start of the Neoproterozoic, but its identity as an animal is heavily contested. 5. Nearly all modern animal phyla first appeared in the fossil record as marine species during the Cambrian explosion, which began around 539 million years ago, Maya, and most classes during the Ordovician radiation 485.4 Maya. Common to all living animals, 6,331 groups of genes have been identified that may have arisen from a single common ancestor that lived about 650 Maya during the Cryogenian period. Historically, Aristotle divided animals into those with blood and those without. Carl Linnaeus created the first hierarchical biological classification for animals in 1758 with his Systema Naturae, which Jean-Baptiste Lamarck expanded into 14 phyla by 1809. In 1874, Ernst Haeckel divided the animal kingdom into the multicellular metazoa, now synonymous with Animalia, and the protozoa, single-celled organisms no longer considered animals. In modern times, the biological classification of animals relies on advanced techniques, such as molecular phylogenetics, which are effective at demonstrating the evolutionary relationships between taxa. Animals have several characteristics that they share with other living things. Animals are eukaryotic, multicellular, and aerobic, as are plants and fungi. Unlike plants and algae, which produce their own food, animals cannot produce their own food, a feature they share with fungi. Animals ingest organic material and digest it internally. Animals have structural characteristics that set them apart from all other living things. Cells surrounded by an extracellular matrix composed of collagen and elastic glycoproteins. Motolicia blastula stage during embryonic development, i.e. able to spontaneously move their bodies during at least part of their life cycle. Cell junctions, tight junctions, gap junctions, and desmosome tissue muscles, which enable locomotion, and nerve tissues, which transmit signals and coordinate the body. Typically, there is an internal digestive chamber with either one opening, in Ctenophora, Cnidaria, and flatworms, or 2 openings, in most bilaterians. Animal development is controlled by Hox genes, which signal the times and places to develop structures such as body segments and limbs. During development, The animal extracellular matrix forms a relatively flexible framework upon which cells can move about and be reorganized into specialized tissues and organs, making the formation of complex structures possible, and allowing cells to be differentiated. The extracellular matrix may be calcified, forming structures such as shells, bones, and spicules. In contrast, the cells of other multicellular organisms, primarily algae, plants, and fungi, are held in place by cell walls, and so developed by progressive growth. Animals are categorized into ecological groups depending on their trophic levels and how they consume organic material. Such groupings include carnivores, further divided into subcategories such as piscivores, insectivores, ovivores, etc., herbivores, subcategorized into folivores, graminivores, frugivores, granivores, nectarivores, algivores, etc., omnivores, fungivores, scavengers/detritivores, and parasites. Interactions between animals of each biome form complex food webs within that ecosystem. In carnivorous or omnivorous species, predation is a consumer-resource interaction where the predator feeds on another organism, its prey, who often evolves anti-predator adaptations to avoid being fed upon. Selective pressures imposed on one another lead to an evolutionary arms race between predator and prey, resulting in various antagonistic/competitive coevolutions. Almost all multicellular predators are animals. Some consumers use multiple methods. For example, in parasitoid wasps, the larvae feed on the host's living tissues, killing them in the process, but the adults primarily consume nectar from flowers. Other animals may have very specific feeding behaviors, such as hawksbill sea turtles, which mainly eat sponges. Most animals rely on biomass and bioenergy produced by plants and phytoplanktons, collectively called producers, through photosynthesis. Herbivores, as primary consumers, eat the plant material directly to digest and absorb the nutrients, while carnivores and other animals on higher trophic levels indirectly acquire the nutrients by eating the herbivores or other animals that have eaten the herbivores. Animals oxidize carbohydrates, lipids, proteins, and other biomolecules, which allows the animal to grow and to sustain basal metabolism and fuel other biological processes such as locomotion. 55. Some benthic animals living close to hydrothermal vents and cold seeps on the dark sea floor consume organic matter produced through chemosynthesis, via oxidizing inorganic compounds such as hydrogen sulfide, by archaea and bacteria. Animals evolved in the sea. Lineages of arthropods colonized land around the same time as land plants, probably between 510 and 471 million years ago during the late Cambrian or early Ordovician. Vertebrates such as the lobe-finned fish Tiktaalik started to move onto land in the late Devonian, about 375 million years ago. Animals occupy virtually all of Earth's habitats and microhabitats, with faunas adapted to salt water, hydrothermal vents, fresh water, hot springs, swamps, forests, pastures, deserts, air, and the interiors of other organisms. Animals are, however, not particularly heat tolerant. Very few of them can survive at constant temperatures above 50 degrees Celsius, 122 degrees Fahrenheit, or in the most extreme cold deserts of continental Antarctica. The collective global geomorphic influence of animals on the processes shaping the Earth's surface remains largely understudied, with most studies limited to individual species and well-known exemplars. The blue whale, Balænoptera musculus, is the largest animal that has ever lived, weighing up to 190 tons and measuring up to 33.6 meters, 110 feet, long. The largest extant terrestrial animal is the African bush elephant, Loxodonta africana, weighing up to 12.25 tons and measuring up to 10.67 meters, 35.0 feet, long. The largest terrestrial animals that ever lived were titanosaur sauropod dinosaurs such as Argentinosaurus, which may have weighed as much as 73 tons, and Supersaurus, which may have reached 39 meters. Several animals are microscopic. Some myxozoa, obligate parasites within the cnidaria, never grow larger than 20 micrometers, and one of the smallest species, Myxobolus shekel, is no more than 8.5 micrometers when fully grown. Evidence of animals is found as long ago as the Cryogenian period. 24-isopropylcholestane, 24-IPC, has been found in rocks from roughly 650 million years ago. It is only produced by sponges and pelagophyte algae. Its likely origin is from sponges based on molecular clock estimates for the origin of 24-IPC production in both groups. Analyses of pelagophyte algae consistently recover a Phanerozoic origin, while analyses of sponges recover a Neoproterozoic origin, consistent with the appearance of 24-IPC in the fossil record. The first body fossils of animals appear in the Ediacaran, represented by forms such as Charnia and Sprigina. It had long been doubted whether these fossils truly represented animals, but the discovery of the animal lipid cholesterol in fossils of Dickinsonia establishes their nature. Animals are thought to have originated under low oxygen conditions, suggesting that they were capable of living entirely by anaerobic respiration, but as they became specialized for aerobic metabolism, they became fully dependent on oxygen in their environments. Many animal phyla first appear in the fossil record during the Cambrian explosion, starting about 539 million years ago, in beds such as the Burgess Shale. Extant phyla in these rocks include mollusks, brachiopods, onychophorans, tardigrades, arthropods, echinoderms, and hemichordates, along with numerous now extinct forms such as the predatory Anomalocaris. The apparent suddenness of the event may however be an artifact of the fossil record, rather than showing that all these animals appeared simultaneously. That view is supported by the discovery of Aurora lumina Atemberufii, the earliest known Ediacaran crown group Cnidarian, 557 to 562 Maya, some 20 million years before the Cambrian explosion, from Charnwood Forest, England. It is thought to be one of the earliest predators, catching small prey with its nematocysts as modern cnidarians do. Some paleontologists have suggested that animals appeared much earlier than the Cambrian explosion, possibly as early as 1 billion years ago. Early fossils that might represent animals appear, for example, in the 665-million-year-old rocks of the Trezona Formation of South Australia. These fossils are interpreted as most probably being early sponges. Trace fossils such as tracks and burrows found in the Tunian period, from 1 Jaya, may indicate the presence of triploblastic worm-like animals, roughly as large, about 5 mm wide, and complex as earthworms. However, similar tracks are produced by the giant single-celled protist Gromia spherica, so the Tunian trace fossils may not indicate early animal evolution. Around the same time, the layered mats of microorganisms called stromatolites decreased in diversity, perhaps due to grazing by newly evolved animals. Objects such as sediment-filled tubes that resemble trace fossils of the burrows of worm-like animals have been found in 1.2 Jaya rocks in North America, in 1.5 Jaya rocks in Australia and North America, and in 1.7 Jaya rocks in Australia. Their interpretation as having an animal origin is disputed, as they might be water escape or other structures. The siphonophores, Siphonophori, are an order of colonial and free-swimming cnidarians belonging to the class Hydrozoa, whose colonies consist of hundreds to thousands of polyps. The individuals within the colony are so highly specialized and diverse in form that they can almost be considered as organs of a single organism. There are feeding polyps, gastro-/autozooids, sensory polyps, defensive polyps, dactylozoids, reproductive polyps, gonozoids, covering polyps, phyllozooids, along with the upper float, pneumatophore, and numerous swimming bells, nectophores. The most well-known siphonophore is the Portuguese man-o'-war, Phasalia physalis. The tentacles of some species can reach up to 50 meters in length. In 2005, U.S. researchers collected specimens of a new species within the genus Arena, Arena spnov, from depths between 1,600 and 2,600 meters, and found that newly formed capture arms emitted bluish-green light while mature tentacles emitted red light. The researchers hypothesized that, contrary to previous assumptions, the jellyfish used this red light to attract rare fish in the deep sea as prey. Siphonophores primarily feed on fish, and 2 of the specimens collected had just digested fish. Since the probability of such random prey encounters in the depths is very low, this could mean that such fish can perceive and be successfully attracted by the red light. The Portuguese man-o'-war, Phasalia physalis, also known as Atlantic Portuguese man-o'-war, floating terror, or bluebottle, is the only species in the genus Phasalia, which belongs to the siphonophores, Siphonophori. A smaller form is occasionally referred to as Phasalia utriculus, but this additional subdivision is not accepted by Worms, World Register of Marine Species. The tentacles can cause welts and typically intense pain upon contact with humans. Although the external appearance of the Portuguese man-o'-war strongly resembles a jellyfish, it is actually composed of an entire colony of interdependent polyps. A variety of polyps join together, each specializing over time in specific tasks like feeding, reproduction, defense, or the formation of capture threads. This ultimately leads to the individual polyps becoming incapable of surviving independently, relying entirely on their union with other polyps. The Portuguese man-o' war is a typical species of pliston, organisms that drift on the water's surface and are displaced by the wind. It is primarily found in the Pacific Ocean but also near the Canary Islands and Portugal. It is prevalent in the Caribbean, such as off the coast of Cuba. In early May 2009, specimens appeared on the coasts of the Spanish Balearic Islands Majorca and Formentera for the first time in a decade. The island residents call them Carabella portuguesa or Botella azul, blue bottle in English. There have also been sightings in Malta in 2001, June 2010, and May 2018. In August 2010, clusters of Portuguese man-o' war were observed along the northern Spanish Atlantic coast in the Basque Country and Cantabria. In April 2018, several specimens were spotted on the beach of Migjorn, southern coast, of Formentera, leading to the beach being closed to swimmers. In the spring of 2019, Portuguese man-o' war were also seen in Madeira and the Azores, and in October of the same year, even off Kerry in Ireland. The main reasons for the increased occurrence of jellyfish in general are overfishing of tuna, rising ocean temperatures, and greater food availability due to eutrophication, which leads to increased algae growth. The bluish shimmering, sac-like gas bladder, pneumatophore, measuring up to 30 centimeters, provides buoyancy. It is filled with a gas mixture consisting of diffused ambient air through the skin and carbon monoxide produced by a gas gland from the alpha-amino acid L-serine. The proportions of the gases vary depending on the location of the investigated specimens. Oxygen content ranges from 13 to 20%, and carbon monoxide content from 0–13% in their native subtropical habitat. Compared to cooler regions, the Portuguese man-o'-war's gas bladder exhibits higher variability. In case of danger, the creature can empty its gas bladder and submerge within seconds. Its sail-like crest is raised only in windy conditions, otherwise, the Portuguese man-o'-war would dry out. While drifting, it repeatedly tilts from side to side to keep itself moist. By controlling its tentacles, it can maneuver just enough to ensure that large groups of several thousand specimens remain together. The numerous blue, white, or reddish-purple tentacles can extend up to 50 meters in length. The tentacles contain up to 1,000 cnidocytes per centimeter, each carrying a venomous mixture of various proteins. This venom acts directly on nerve cells upon contact with skin, leading to overstimulation. It can kill small fish and other prey. For humans, stings cause intense pain, leaving red welts on the skin resembling whip marks. The welts typically fade after 2 or 3 days, and the pain subsides within about an hour. However, the venom can also reach lymph nodes, causing greater pain. A healthy adult can endure burns from the jellyfish without life-threatening consequences. For weakened individuals or allergy sufferers, there's a risk of allergic shock, which can be fatal. Fatalities are extremely rare and are sometimes misattributed to encounters with the much more dangerous box jellyfish. Medical advice recommends consulting a doctor if pain is severe or persistent, if wounds worsen, or if symptoms of illness or inflammation occur. Detached tentacles still contain active cnidocytes. These cells remain dangerous for several days, even when jellyfish are washed ashore. Some medical sources advise against washing stings with vinegar or fresh water, while others suggest vinegar to soothe the skin. In any case, it's recommended to carefully remove tentacle remnants with seawater, avoiding direct contact with the tentacles as they can continue stinging. Hot water above 45 degrees Celsius (113 degrees Fahrenheit) can denature the venom's proteins. Treatment typically involves zinc gluconate. Despite its venomous cnidocytes, the Portuguese man-o'-war has natural predators. These include the loggerhead sea turtle, Caretta caretta, the pelagic nudibranch Glaucus atlanticus, and the violet snail, Janthena janthena. Ocean sunfish, mola mola, also consume Portuguese man-o'-wars. The shepherd fish or jellyfish fish, Nomius granovii, is partially immune to the venom of the Portuguese man-o'-war. It lives among its tentacles, avoids the larger ones, and feeds on the smaller ones below the gas bladder. In doing so, it may also attract other fish that serve as prey for the jellyfish, creating a symbiotic relationship. The bar jack, Carangoides bartholomeyi, also known as yellow jack, is a moderately popular food fish that can itself be toxic, due to ciguatera toxin. It frequently acts as a symbiont around the West Indian Islands, residing among swarms of Portuguese man-o'-wars. That's all for now. In the next episode, we'll dive deeper into Golem and its connection to Blastoise. Don't forget to subscribe, leave a like, and join us next time.